asked to talk about potential opportunities for clinical trials. And I am going to focus on TKIs. And I'm an investigator on the Lenvatinib trial that is actually in combination with chemotherapy in its current incarnation. So again, kind of following on from what we've heard about the relative lack of progress, I guess a key question is, how much signal is enough to move forward into first-line therapy? And that's a slightly philosophical question. I have a view, but I guess we need to have a consensus. Slightly different way of looking at what progress has been made. This is a review of 99 studies over a long period of time in refractory relapsed osteosarcoma. Again, demonstrating fairly similar things to the 7 COG studies, that we've tried a lot of different agents in combination sometimes in these patients where disease has got big and strong and come racing back, and those therapies have not been very effective. So we've got 99 studies, overall very limited progression or partial response or complete response. Although osteosarcoma, that's quite a difficult thing to measure. Our tumours don't necessarily shrink. We're mostly measuring these things by RECIST, and it's a tough call. So indeed, progression-free survival is perhaps a better proxy, and the better numbers on this are indeed predominantly TKI studies. But none of these agents have made it into first-line practice. They've been tested and they haven't come through. So a little bit about tyrosine kinase inhibition. I'm not a scientist. I'm not going to tell you chapter and verse on this. But just to say, a picture of the human kinome there. Wow, that's complex. Wow, there's lots of kinases there, and they do just about everything in the cell. And we talk about these as targeted agents. They are small molecules that fit into the kinase domain, more specifically for some kinases than others. But if you give the right concentration of them, they will hit on lots of these kinases. So they're doing lots and lots of different things. There are some very targeted situations. So if you have a driver mutation and you block that mutation, then in BCR-ABL leukemia, you will turn that off. In NTRK fusion-driven sarcoma, you'll turn it off. But that's not really what we're doing with these drugs in osteosarcoma, usually. So from phase 1s that have been done for kind of broad patients, you sometimes see responses in osteosarcomas, and that's driven a bit of this process. But this cartoon with your effector and your downstream, this is such a simplification that it's almost grotesque. And so the kinases that the TKIs that we're mostly talking about can be represented in these sort of balloon plots, and they're mostly driven and drawn to show differences, but you can also see a lot of similarities here. And the question of which drug, if we thought we were going to use a TKI for osteosarcoma, we should use, is a very difficult one, and I don't propose to answer that just now. And just as a slight point, we often are talking about these as VEGF inhibitors, but when St. Jude's did actually test a different way of blocking VEGF with bevacizumab, that was a negative study. That didn't produce any change in outcome for patients. So I'm going to focus a little bit more on the actual studies. So there's been 2 studies out of the Italian group. First of all, with single-agent Sorafenib, which produced this sort of survival curve. And again, it's a number above the 40% progression-free survival. The responses are sustained up to about a year for some patients, most patients not. The group tried to augment that outcome. They had some preclinical data suggesting that mTOR inhibition might assist that, and so they put an mTOR inhibitor in. it didn't make their target for being effective enough. They'd set a 6-month 50% progression-free survival, which they didn't quite make. There perhaps was a slight difference, although these are not strictly comparable studies. A newer presentation from a Chinese group looked at Apatinib, which is said to be a narrower spectrum TKI. Again, I'm not enough of a chemist to say for certain that that's absolutely the case. What's interesting, I think, about Apatinib was they seem to actually get quite a lot of objective responses, which is fairly unusual. So the waterfall plot here shows a lot of tumours actually diminishing in size, which is pretty unusual in osteosarcoma. We haven't seen that very often. They had quite a lot of pneumothoraxis, which is a toxicity that has occurred in a few of the other studies, not to this sort of extent. But the responses were actually very short-lived. So they got shrinkage, but then the tumours rebounded rapidly and grew. Regorafenib study has actually tested whether this is truly an effect much better. They did a randomized study where they randomized 2-to-1 against placebo. The placebo line is this blue one here, and this is what probably truly happens with osteosarcoma when it's refractory and relapsed and you don't do anything with it. They progress extremely quickly, which makes the difference that you see with Regorafenib rather more striking. And again, the waterfall plot not quite as impressive as Apatinib, but still there are some actual objective responses. And so this is the French Regobone study. The Regosarc trial in the US was running kind of concurrently, but published or is reported slightly after, but says pretty much the same thing. And when they took the patients who'd progressed on placebo and crossed them over, they did something quite similar to the patients who'd originally had Regorafenib. So you got a similar sort, it's not statistical, there's only a small number of patients, but it looks actually very similar, and probably the same thing is going to go on. So the survival swimmer plots show that it took a while to progress once they changed over, and there were some responses. I've been involved in the Lenvatinib study, which was originally a pediatric phase 1, and slightly different to the other studies. This is predominantly a pediatric study. So the original phase 1 was about dose finding, and the dose found was basically the same as you would predict for adults. The study hasn't reported, but all the data that I'm sharing here is in the public domain from presentations that have already happened. And then the phase 2 expansion was both a single agent and then in combination with chemotherapy. So the progression-free survival looks a little bit less disappointing than the other TKI studies, but it's not a direct comparison, and it's a relatively small number in this study. The toxicities were really as expected. There were some actual partial responses, and we moved on to a chemotherapy combination study. And the chemotherapy combination study isn't complete. There's 3 more patients required to complete that, but it should complete this year, and we're hoping to present it possibly at SIOP later in the year. The toxicities really are as you'd expect. It's, I'm going to say, feasible and possible to deliver this drug with ifosfamide and etoposide. We've certainly done a number of those patients in my centre. Interesting, maybe, you see a funny vacuolation of lung mets. I'm not sure it's important, but it's striking in pictures. And what I think we're finding is that the progression-free survival is, for some patients at least, substantially prolonged. So I've got a number of patients who are a couple of years on this therapy without progression. There is a PIP for Lenvatinib, with an intention to proceed next to a randomized study in relapse between Lenvatinib and chemo alone. So that's some TKI data. What should we do with it? I would say, to my mind, that there's an abundant evidence that tyrosine kinase inhibitors have biological activity and that they prolong survival. I would say that they've only been tested in situations where the disease has got big, has got strong, has overcome its microenvironment starting points, and maybe is not the best place for a drug like this to be used, but yet has still shown benefits. So I think that it should be brought frontline or into occult disease. I don't know which drug. I think there's a couple of different time points that you could look at this at. Certainly, obviously, you could use them as in combination with chemotherapy at the outset of treatment. But I would argue you could also consider at the end of standard chemotherapy, when you have debulked the patients that are going to progress, where you have a minimal disease state, that you could use these drugs and maybe get more benefit than we've seen in the studies where we've used them. So in upfront therapy, you'd have the opportunity to target naive tumour. You could combine with the most effective chemotherapy. We've only used it in the Lenvatinib study alongside ifosfamide and etoposide at a relatively low dose. But we have demonstrated, I think, that TKIs can be tolerable with chemotherapy, and there are a number of situations where TKIs have been given with a range of chemotherapies, not necessarily the ones that we're talking about. You would need dose finding with standard MAP. Many of the TKIs decrease cardiac measured function, and that could be a problem with your anthracyclines. And you've got your question about interruption for surgery. Which patients we'd give it to? It's a different question. Thank you. And the other alternative that I would see is that you could use it again, as I say, as single agent after initial therapy. In some ways this would be easier. We know what dose we would use. We would have no competing studies. What might be a problem is that patients are often sick of being treated by this point. They might not be keen. Which drug would we use? Again, I'm not saying there's an answer to that exactly just now. And how long would you give them this treatment for if you were going to do it? Should we randomize everybody? Should we say that metastatic patients do so badly that they should just receive treatment and we could compare with historical controls? These are questions that I'm not answering just now. That's me.